Good afternoon, everybody. It's 12 noon, and we are starting, hard to believe, our last session in the mini-series, in the first mini-series for our department, Molecular Pathology for Surgical Pathologists. And I just want to remind everybody to mute your microphones because we have a large number of participants. Please ask your questions in the chat. They will be answered as we go by one of the presenters, as well as we will have some time at the end of the session. And I want to remind everybody to fill out all the evaluation forms that we send you. It's extremely important, especially because of our inaugural event. We want to understand how that format works for you. Was the topic relevant? And if you have any suggestions for us, we read all the forms. With that, I would like to welcome again Dr. Yael Fisher and Dr. Elan Hahn to present today. In my understanding, Dr. Hahn, it's your turn to present.
Yeah, thank you very much for the introduction. Thanks everybody for coming to the course and sticking through till the end. I have no relevant financial relationships to disclose. So first, what we'll do today is go through a little bit of a review from last time, and then we'll be moving on to variant classification and the two different classification schemes. We'll go through useful databases, for which there are many. Move on to the Ontario Health Cancer Care Ontario coverage and requirements for specific testing indications, and end with some illustrative cases. So first, to start with some review. So this is what Dr. Fisher went over in the first session. DNA is made by base pairs of nucleic acids, and A and T always pair up, and G and C always pair up with different amounts of hydrogen bonds between them. The central dogma of molecular genetics is that DNA is transcribed to RNA, which is then translated to protein, which will then take on the effect in the cell. And it's this impact on protein that we're really looking for, and we're trying to infer from the DNA. And we could look for multiple different types of alterations. Copy number changes, which we have gains or loss of segments of the DNA, and we could look for this through next generation sequencing, through FISH, through chromosomal microarray. So there are multiple different ways we could look for copy number variation. Next, we have gene fusions, where we have parts of one gene connect to parts of a different gene, which impacts function when we report it and when we're looking for it. And this impact on function is what is driving, or is what is contributing to the disease state. Could be done by DNA or RNA sequencing, with most labs using RNA sequencing to cut out the variable and link the intronic component, so that we're just looking at exons, which will be easier for us to detect. And also incorporating expression, which is the amount of RNA being made. That tends to contribute to the oncogenicity of the fusions. And there are many different types of genomic alterations that include single nucleotide variants that could be missense, that will change the amino acid, or nonsense that will result in termination, or some that impact the splice site. And we also have deletions or insertions, which could remove or insert certain amounts of DNA. And these could either be in frame, which would maintain the reading frame, or cause a frame shift, which will cause an early termination. And then we went on to cover, specimen selection, and each platform or assay will have its own cellularity content that is needed for the assay to be successful, or what it's been validated on. So we want to make sure we're not getting too much necrosis, which will have a lot of nucleases, and we also want to make sure we're not having too many interfering normal cells, like lymphocytes, as you can see in panel C, and that we have enough tumor cells, and that's sort of borderline on panel D. So once we get our specimen, we extract the nucleic acid. So we lyse the cells, we clean up, remove some of the contaminants, purify for the nucleic acid that we're looking for, wash off or wash out whatever waste there is, and then elute our purified nucleic acid. And then we'll be looking into the quality and quantity of what we've eluted. So on the left is that PCR, quantitative PCR method to look at the length of fragments that we have. And on the right is the DV200 length of, amount of RNA fragments we have that are over 200 nucleotides. And those are very important quality metrics to look at from your extraction to forecast downstream what quality we're expecting out of the sequencing. And there are different types of sequencing. So there's PCR, polymerase chain reaction, which is multiple cycles of denaturation, annealing, and extension, which can be quantified as well, looking for the production of fluorescent signal that increases as we increase the sequencing and amount of our target sequence. And then there's Sanger sequencing, which you have varying lengths of increasing lengths of fragments within our sequence, each signified by a fluorescent probe. And then reading the fluorescent probe of the different lengths segments, we could rebuild our sequence, as you can see under number 3. And then we move on to next generation sequencing, where we essentially do the sequencing massively in parallel. So there are 2 main approaches: amplicon-based, which are amplifying targets of to our target regions, and hybridization capture, which we select out by hybridization to specific probes. And then there are the 2 main chemistries, which is Illumina, where we create this lawn and have the bridge amplification, and then look at the light signals based on the fluorescent nucleotide that was incorporated. So we take multiple of these pictures through every wash cycle, and then build the sequence that way. And the other is the semiconductor sequencing, which is the Thermo Fisher ion torrent chemistry, where we wash over a nucleotide at a time, and we see if there's hydrogen released, and we measure that pH change. And if we see a pH change, we know that that specific nucleotide that we washed across was added. And once we get all of that data, we create a FASTQ file, which is not aligned yet. And then once we take those reads, and each of those entries is a read with a quality metric, and we take that and we align it to the reference sequence that creates our BAM file. And then once we're calling variants or differences from the reference sequence, that creates our VCF or variant calling file. And then once we start to annotate our variants, this is what it looks like here, where you have your chromosome position on the chromosome, the reference sequence, the alteration sequence, and then the allele frequency. The depth of coverage at this position specifically, the gene involved, and then we have different ways to relay the nucleic acid change. So that's the codon change or the amino acid change. So if you see HGVSC, HGVSP, so those columns are the codon change and the amino acid change or the protein change. The variant type, our RefSeq transcript ID, which is very important to record so that we know exactly what position we're looking at when trying to compare reports to from different labs. And then some other database information, which could be publicly available databases, as they are in this case. So ClinSig is pulled from ClinVar, and the cosmic count is amount of these variants in tumors and cosmic, and we're going to get to those databases in a little more detail today. And then some prediction tools to see, is it predicted to be deleterious or not? So just to give an overview of this, of the pipeline, we extract, prep the library, sequence, call our bases at each position, align them, and then see how that difference differs from the reference sequence and call our variants. And now we're going to talk about interpreting those calls. So the general approach here is to see, number one, is it technically real? And number two, is it functionally impactful? So technical assessment, it's a lot of what we spoke about previously. Um, there are pre-analytical factors like the sample type, the fixation used, the fixative used, the length of fixation, the cellularity of the specimen. Because let's say you see a variant allele frequency of 3%, but your tumor cellularity at the time of circling, the pre-analytical factor of cellularity was much higher, was 80, 90%. That's going to make you a little more suspicious of that call as opposed to if it was a very low cellularity specimen. Other analytical issues, common artifacts like complex insertion deletions are hard for bioinformatics softwares to call specifically. Then we have assay-dependent issues that include homopolymer or amplicon end in an amplicon-based sequencing, for example. Those are a little more difficult to call. And post-analytical would be, copying that information out of your output and putting it into your report or misclassifying a variant, for example. So we're going to touch on this again a little later, but this is my general approach to how I look at these cases is I look at the variant allele frequency of the called variant, and that I take into account the cellularity of the specimen, but mostly I take into account what our platform is validated to, what's the lower limit of detection in terms of variant allele frequency. Then I look at the automated information from ClinVar, and this is a germline enzymatic database, and we're going to get into it a little more later today, and see which of these were called pathogenic or likely pathogenic, and look into those variants. Next, I'll move to the variant type. Is it in the intron? Is it upstream in the untranscribed region? Is it a missense, null variant? So is it synonymous? So these types of variant types and changes will have different implications in different genes. Like a nonsense variant, for example, in a tumor suppressor gene will obviously hold a lot more weight than the same thing in a proto-oncogene. Then I go on to see how many of these are known benign in public databases. I look in NOMAD, which is a population database, to see the minor allele frequency in the population at large. And if it's higher, then I'm more suspicious of those variant calls. I'll look in our in-house database to see if there's anything specific in-house that would cause this to cause a certain variant to come up more often, which would make me think it's more likely an artifact, if it's not clearly pathogenic. And then once I whittle it down, I interpret those relevant genes. So first is the functional assessment. So once we get done with our technical assessment, and now we're moving on to our functional assessment, we could split it up into, two, I think, large domains. One is the functional impact on protein function. So oncogenicity is how that's been described, and clinical significance. And these are the tiering from the AMP-ASCO-CAP guidelines. So I'd like to add a brief word about germline versus somatic variants. In essentially all cases, the underlying molecular mechanism of the same variant in germline and somatic contexts is the same, but the implications of the same variant in germline and somatic contexts could be different. For example, BRAF V600E, and I've used this mutation, as you'll see going on in this presentation, has oncogenic effects and therapeutic implications in cancer, but in germline, it really presents with embryonic lethality. Another example is variant types within a single gene, somewhat common in proto-oncogenes. So in sporadic malignancies, oncogenic TERT alterations, specifically TERT promoter mutations or copy number gains, lead to an increase in TERT expression, but in the germline context, pathogenic TERT alterations are loss of function events. And based on the current body of evidence, such alterations fall under the VUS umbrella in somatic context. Germline pathogenic variants in hereditary cancer genes have lifelong implications for affected individuals and also have potential implications for their families. And when we find them in the Semitic setting, we may be suspicious of their germline origin due to factors like clinical history or allele frequency, which can range due to specimen characteristics or copy number changes or loss of heterozygosity. But in the absence of germline testing, even with a family history and a high allele frequency, we still can't be certain that they're not only present in the tumor. So, in a number of instances, there are significant differences in clinical implications depending on if it's somatic or germline. So, in such cases, germline testing should be considered to confirm germline or somatic origin. Additionally, some variants in hereditary cancer genes may be present as germline but not as somatic, and vice versa. So, there are a few possible explanations for that observation. It could be variant genomic location and nucleotide composition around such locations may be more susceptible for particular types of mutations. And it may only be relevant in the embryonic or postnatal context. Also, some variants in tumor suppressor genes may have particularly strong dominant negative effects and may result in embryonic lethality. And for a lot of these variants, ClinGen variant curation expert panels have been formed and developed, and they've made these gene-specific guidelines through modifications to the ACMG AMP germline pathogenicity guidelines. So moving towards the workflow in looking at any variant. So we identify these variants through variant calling. We annotate them, and then we look at determining, are these functionally relevant? Once we determine that they're technically real, are these functionally relevant? And that's where oncogenicity classification comes in. So these guidelines were made by the ClinGen, CGC, and VIC. And they define oncogenicity as diagnostic, prognostic, and therapeutic information from molecular pathology. And all of that information really is built on the foundation of whether or not this variant is oncogenic. It was preceded by the AMP-ASCO-CAP tiering, which I think theoretically or logically should follow oncogenicity. So once we determine that it's functionally relevant, then we could start to talk about its clinical significance. And in those guidelines, there was no real systemic standards to classify the functional impact of molecular findings. So pathogenicity of the variant in the context of neoplastic disease is the goal of these guidelines. It's the mutation's ability to cause growth and survival advantages in tumor cells. And it's not that these guidelines and the AMP-ASCO-CAP guidelines are exclusive of each other, but in an ideal world, they would be used together. So oncogenicity, it's very rigorous, and they give point ranges for each classification. So you range from benign, likely benign, to variants of uncertain significance, to likely oncogenic and oncogenic. And we're going to go through the rubric now. So very strong is that OVS1, and this would be a null variant in a bona fide tumor suppressor gene. So if it knocks out the function of the tumor suppressor gene, we're pretty sure that it is going to be oncogenic. It can also be used for splice variants up to -16 at the splice acceptor site or +5 at the splice donor site if there's experimental evidence indicating splicing effects. And there's a recent created resource called MuteSpliceDB that provides a place for the genomic community to catalog RNA-seq-based evidence for splice variants effect on splicing. So if you have RNA-seq on a sample that you have, a variant that seems to be at a splice site and you're not sure if it's impacting splicing, you should actually be able to see the splicing effect in the RNA data. And that's one of the ways that RNA data is being used clinically, in addition to the main way, which is fusion detection. So otherwise, being present in hotspots is also something that we look into. And just a word about tumor hotspots. So on the top, PIK3CA and IDH1, these are proto-oncogenes. And these mutations in proto-oncogenes, in order to cause cancer, need to be in very specific locations. As you can see, they have a lot of missense mutations in very, very specific locations. They need to be in specific domains that will actually have a functional impact. So that's where you get these hotspots from. And RB1 and VHL are tumor suppressor genes. And really what you need to do is sort of impact the function or knock out the function of these genes. And while there are still hotspots in RB1 or VHL, the spread of these mutations that are pathogenic, that knock them out, in databases is much more even, because it's a lot easier to knock out the function of one of these genes by mutating it anywhere than it is to activate a proto-oncogene. So OM1 would be that it's located in a critical and well-established part of a functional domain. And we spoke about hotspots on the previous slide, and it brings up a point that some of these criteria may have potential overlap, which could lead to double counting. So there are explicit exclusions specified here in this table. For example, OM1, it's based on variants being located in critical and well-established parts of functional domains. And it's noted that it can't be used if OS3 is applicable, because OS3 from the previous slide is based on a variant being in a known cancer hotspot. And such hotspots are frequently located in critical functional domains. So to avoid that double counting, we have that exclusion. So OM2 is protein length changes as a result of in-frame deletions or insertions in a known oncogene or tumor suppressor gene, or stop loss variants in known tumor suppressor genes. OM4 is missense variant and amino acid residue where a different variant is oncogenic. So this is important to note that it's really when these amino acid changes, the difference in the amino acid is greater or at least or similar to the one that's documented as oncogenic. So, and that's important because you don't want potential misuse of this criterion in cases where the substitution is actually for an amino acid with a very similar physical chemical property. There are some well-known amino acid difference metrics, such as Grantham's distance, Epstein's coefficient, or Meatta's distance, that can be looked into. There's a generic example here that they describe. So it's the arginine 156 to his. So the chain from arginine to histidine is known to be oncogenic. And then you find arginine to cysteine. So looking into it, we need to see, is the difference between cysteine and histidine greater, between cysteine to arginine greater than that of histidine to arginine? And I think there's a good, this is what some of those distances and coefficients are looking at, and it's the properties of the amino acids. So as you see, we change from A to B, and that's considered oncogenic. Since we're changing from A to C, and since it's much more different physicochemically, we could say that it satisfies that criterion as being the same amino acid, a different amino acid as a known oncogenic variant. But if it were more similar in its physicochemical properties to arginine, then we wouldn't be able to use that criteria. So now these are, we're going in decreasing strength of evidence. So OP1 here is all utilized lines of computational evidence support an oncogenic effect of a variant. And these are in silico algorithms that we're going to get into a little more later. Somatic variants in a gene in a malignancy with a single genetic etiology. And then we have our hotspot criteria, but here again, now we're going even lower in the count, where in the first case, the instances in cancer hotspots needed to be higher. Then at the next level of evidence, OM lower, and now even lower for OP3. And OP4 is absent from controls in population databases like NOMAD. And then we move to benign categories. So benign evidence. The top one, very strong benign evidence, is that it's present in more than 5% of any of 5 general continental populations. And then we go down more nomad evidence. Then we have in vitro or in vivo functional studies that show that there are no oncogenic events. And then we go to in silico evidence again, and then a synonymous variant for which splicing prediction algorithms predict no impact to splice consensus, nor the criterion of a new splice, and the nucleotide is not highly conserved. So, but this is again is just a supporting point, which would be -1 point. And when you put that all together, you're looking for a score of 6 or more to be likely oncogenic or oncogenic. And generally thinking about this data, population data is very important. So population data is, one of the main theoretical things that we use to determine the oncogenicity of a variant or the functional impact of a variant. Then there are well-established functional studies that we would look for. Predictive data, which would be in silico, cancer hotspots, which again is another a very important resource, and it's logically one of the important things used in this classification. So between population resources and cancer hotspot resources, you could do a lot of the classification according to this rubric. So we'll go through one example here, trying to apply this rubric very rigorously. So BRAF G469E. So there are functional studies that show low BRAF kinase activity in this variant. So that's a functional impact. So that would be OS2, well-established in vitro or in vivo functional studies supportive of an oncogenic effect of the variant. And just in general, with BRAF variants, we classify them into 3 classes, whether or not they're dependent on dimerization or RAS. So wild type needs both. Class 1 BRAF mutants like D600E function as monomers and independent of RAS. Class 2s need to be dimerized but are independent of RAS, and class 3s need both. In silico pathogenic deleterious predictions for this variant, so that would be OP1, that all computational evidence supports an oncogenic effect. Cancer hotspots has a count of 4, so that's OP3 because the count is below 10. And it's absent in the population controls, which will give us 1 point. So altogether, that's 7, and that puts us at likely oncogenic. So now we'll move on to determining the clinical significance of variants that are detected. And this was an article in 2017, which the Association for Molecular Pathology, ASCO, and CAP put together. So to broadly assess the current practice of NGS testing for tumor tissue, technical and reporting groups were surveyed by a survey prepared by the AMP working group. 67 responses were received for the technical survey, 44 for the reporting survey, and most people responded that their panels included up to 100 genes. Most of them detected single nucleotide variants, 95%, 35% detected CNVs, and 37% detected fusions. So there was a lot of variability, which prompted the creation of these standards. So people were reporting down to a minor allele frequency of 3%, some 5%, some 1%. Some used 5 categories of, pathogenic, likely pathogenic, VUS, likely benign and benign. Some 3 categories would be pathogenic, VUS, likely benign. Others had just 2 categories of pathogenic and VUS. Some people reported therapies that were approved by the FDAs and used in professional guidelines, and some did not. Some included the recommendation for genetic testing, and some did not. About 50% of respondents reported allele frequencies, while the rest didn't. Similar findings for genomic coordinates. Transcript ID was a little more reported, but still not always. And sometimes included, only if QC measures were sometimes included only if quality control fails were present in actionable areas or targeted regions. So this was really meant to be an evidence-based categorization of variants looking towards impact on clinical care of these variants. And something would be considered a biomarker if there was evidence of a therapeutic response, if it was an inclusion criteria for clinical trials, if it influenced prognosis or established a diagnosis or warrants establishing surveillance for a potentially germline risk allele or risk gene. So these genes were considered therapeutic, prognostic, diagnostic, or preventative. And it could be different for the same gene in different contexts. For example, IDH1 or 2 variants in gliomas have prognostic significance, whereas in chondrosarcomas, they have diagnostic utility. And in AML, they could be therapeutic. So this is a famous figure, and this is how they tiered variants. Tier 1, variants of strong clinical significance. Tier 2, variants of potential clinical significance. Tier 3 would correspond to VUS, and Tier 4 would be benign or likely benign variants. So they split up the evidence into level A, B, C, and D. So level A evidence is biomarkers that predict a response or resistance to FDA-approved therapies for a specific type of tumor, and biomarkers included in professional guidelines that predict a response or resistance to therapies for a specific type of tumors. Biomarkers included in professional guidelines that are diagnostic and also prognostic were considered level A. Level B would be biomarkers that predicted response or resistance to therapies for a specific type of tumor based on well-powered studies with expert consensus. And that's the same for diagnostic and prognostic significance. It needs well-powered studies with expert consensus. Level C, so this is now moving to tier 2, is biomarkers that predict response or resistance to therapies approved by the FDA or professional societies for a different type of tumor, or biomarkers that serve for inclusion criteria for clinical trials, or are of diagnostic significance based on the results of multiple small studies, or prognostic significance for the same reason. And then level D would be biomarkers that show plausible therapeutic significance based on preclinical studies, or may assist with disease diagnosis and or prognosis themselves, or along with other biomarkers based on small studies or multiple case reports without consensus. So in order to, get the hang of these, that type of significance and this type of analysis, both for functional analysis and for, clinical applicability, there are many databases that are available. So there are population databases, which we're going to touch on NOMAD. There are variant databases, somatic and germline databases, and we're going to touch on some of these, but not all of these. There are protein structure and function databases, genome viewers, gene fusion databases, and other helpful tools that sort of aggregate a lot of these databases together. It's important to understand the content of the database that you're looking at and how the data are aggregated. When you're interpreting these, documentation or published literature relating to a database, it's important to review some of that so that you can know the source type and intent of the database. There are lots of limitations within these databases, so it's important to be aware of these limitations to avoid overinterpretation based on any one of these databases. Very importantly, we need to confirm the annotation. So using genomic coordinates is usually the best way to confirm that you're looking at the same variant that you're seeing. You want to assess the quality of the provided genomic data based on the source from publications or another database, the number of a specific entry, is it single or multiple, the depth of the study, did they use appropriate controls, did they confirm that it was a somatic variant, or is it potentially germline? And we also, when possible, to verify the quality of the pathologic diagnosis provided, if they, talk about the site or the histotype, or if there are images available. So firstly, we could talk about NOMAD, and this is that population database. So this was made without paired normal tissue. The 1% minor allele frequency in the population is used typically as a cutoff of, if you see it above 1%, it's likely a polymorphism. Usually we use the global allele frequency, but it gives ethnicity-specific allele frequencies as well. There are caveats that, insertions and deletions may be underrepresented, partially due to technical challenges associated with their detection. Also, due to clonal hematopoiesis, some variants in NOMAD may actually be somatic and not germline, and when they're in specific genes that are known to be involved in clonal hematopoiesis, it's important to be aware of this phenomenon. You could also examine the read data in NOMAD in their variant report page for very low variant allele frequency variants to maybe help identify what could be caused by clonal hematopoiesis. And some variants are derived from normal tissue in individuals who are known to have cancer, and the non-cancer subset of NOMAD excludes such individuals, so you're able to actually exclude them. So this is what it would look like if we're looking up a specific gene, and for all of these exercises, I've been using BRAF V600E. So here on the top, you can see the variants present in ClinVar. Then next would be the variants reported in NOMAD, and you could toggle which ones you want to look at, synonymous or missense. And looking down, there's no V600E here, and that makes sense, not present in the population. But if we do click on one of these variants, you can see the breakdown based on allele frequency in different populations and a total count as well, which is helpful for interpreting these variants. So then we can move on to somatic or cancer databases. These databases provide information about the incidence and prevalence of variants across the spectrum of different cancers and subtypes. It cross-references to other genomic databases, as well as to published literature, and there's some cellular pathway data, targeted therapy data, and clinical trial and outcome data as well. The prevalence and distribution of sequence variants across different cancers extracted from these databases should be interpreted with caution because of the poor representation of the pathology diagnosis standards. You don't really have that clinical grade literature curation, and it's quite loosely controlled, the sources of the submitted variants in some of these databases. So COSMIC is the first one we're going to talk about. Now you need to register for it. If you register as a researcher, I think it's still free. It provides information on frequency of somatic variants in cancer. It's provided by methods with different degree of genome coverage, and therefore, some coverage across the gene sequences isn't uniform. In some cases, the origin of the variant, germline or somatic, may not be known or available. So when we search for BRAF gene, you have that left-sided menu that you could look at. If you want to look specifically for the variants, click on variants. It will bring you down to a table with all the variants, and you could type in anything in here. This could be the codon location or the amino acid residue. So if I type in V600, the amino acid residue, it takes me to all of the V600, including mutations. And if I want specifically V600E, I put that in, then I would click on it, and it will take me to this page, which is an overview of this specific mutation in COSMIC. It goes through the annotation, talks about drug resistance, and on the left-hand side, you can see it will have different information about the tissue distribution, samples, pathways affected, and references. So tissue distribution, no surprise. BRAF V600E, very common in the thyroid, skin, and large intestine. Then if you're looking at the samples, it will tell you exactly, the sample name and the PubMed ID and the pathways affected, and there are also references lower down on the page. Cancer hotspots is one, that features heavily in the ClinGen paper for oncogenicity classification. It provides information about statistically significant recurrently mutated positions in around 25,000 tumor samples. The determination of the significance takes into account nucleotide mutability and gene-specific SNV rates. It's important to keep in mind that the statistical significance in this resource specifically, it's calculated for the amino acid position and not for the specific change at the amino acid position. It has good coverage for many solid tumors, but it's limited for hematologic cancers and rare and pediatric malignancies. So here again, I'll walk through looking for BRAF V600E. It's there on the main page, but you could search by gene, which will pull up all of the hotspots in a specific gene. And then if you hover over, as I did here, V600E, you could see V600E is the most common change at this hotspot, and you could see the count of different tumor origins that have that specific mutation. So OncoKB, it's a comprehensive, expert-curated precision oncology knowledge base developed by MSKCC, Memorial Sloan Kettering Cancer Center. Systematically annotates both somatic and germline cancer variants, providing clinicians and researchers with evidence-based information on oncogenicity, prognostic significance, and clinical actionability of specific alterations. Each variant is assigned a level of evidence, reflecting its predictive value for drug response, based on FDA and National Comprehensive Cancer Network, or NCCN guidelines and the literature. It's really nice for looking at specific links between molecular changes and therapies. And this is what it would look like here, and we're going to go into it a little more in one of the case presentations, so I will just show you the front page for now. So cBioportal, it integrates large-scale genomic projects, including mutations, copy number alterations, fusions, gene expression, and methylation changes in these projects where available. It's a pretty user-friendly interface for visualization and analysis, and there are link outs to clinical trials and to other resources. And this is what the front page looks once you go in there, and you could choose your studies that you're going to look at, and you could query by gene or just explore those studies fully. And again, we're going to go into this a little more later in one of the cases. So ClinVar is a public database of variants and their clinical significance that's maintained by the NIH. It aggregates and displays variant interpretations submitted by clinical labs, research group, expert panels, and clinicians with level of evidence. So there are different levels of evidence for different entries. So it's very important to be aware of that and look at that because it is very much submitter powered. So you need to look carefully at the date and level of evidence of a submission and use it in conjunction with other resources. Some rare variants may have, an oncogenic entry from a long time ago that hasn't really been confirmed. So this is not to be trusted blindly. It includes both germline and somatic variants, and it integrates with other resources as well. So this is what the front page looks like. You could type the gene into the search bar, and then you'll get the gene. And then you could select and sort the different variants. So here I'm looking for V600E, so I'm looking for a missense variant. So you can see that I clicked that on the left. And you can also see that BRAF V600E pops up, and it has both a tier 1 strong and oncogenic classification here. And if we open up that specific variant, it shows you the different submissions. In the germline, it has conflicting classifications of pathogenicity. Most of them are pathogenic with 1 star, but in somatic, which is what we're talking about today, it's tier 1 and it's oncogenic with 2 stars of evidence. Just looking at a different, and then if you go down, you could see the different submissions, and this is for a germline example of V600E, and you could see what each submitter put as a comment to go along with the variant. So for example, this is what Invitae, or what was formerly Invitae, put in for a germline entry for BRAF V600E. And if we look at a different variant, V600G, this was reviewed by an expert panel, and you could see the level of evidence is 3 stars, which is higher, and it's in an FDA-recognized database, which is through ClinGen, the RASopathy Variant Creation Expert Panel. So this is, a much higher level of evidence than, let's say, a 1 star from 10 years ago. This is a more recent Variant Creation Expert Panel with 3 stars of evidence, which is much more trustworthy. Next, we can move on to gene-specific databases. Like, For BRCA, there's BRCA exchange, which you could search by codon change or amino acid change. And then there's the P53 database, which again, you could search and look through the functional data, tumor variants, germline variants, and there's a lot of information in both of these areas. And then we move on to more of that clinical actionability assessment. And there are different databases for these. So FDA approval for a drug in a certain indication would be something that's associated with a more, with more clinical actionability and AMP Tier 1. NCCN guidelines, National Comprehensive Cancer Network guidelines. So if it's in these guidelines, again, that's enough for Tier 1. And this also, you could just get a membership and for free, and you're able to look through all of these guidelines for each cancer type. The WHO Blue Book, which we're all familiar with, clinicaltrials.gov, which would be a repository of clinical trials that exist, and more locally would be Ontario Health or Cancer Care Ontario guidelines for biomarker testing. And this has, it's just a list of all the different indications in different tumor types, what the targets are, so what you should be looking for testing indications, and what centers perform these tests. So here, for example, there's advanced metastatic breast cancer where directed therapy is being considered, looking for mutations in AKT1, ESR1, PIK3CA, and PTEN, with a list of institutions. And thyroid cancer, all metastatic thyroid carcinomas, including sporadic medullary and radioiodine refractory, well-differentiated thyroid cancer as well. And there we're looking for BRAF, HRAS, KRAS, NRAS mutations, and NTRK and RET fusions. And optional would be a PPR gamma fusion. So those are the more guidelines, clinical actionability assessment. And then moving on to internal databases, these are really helpful when you have a good quality institutional database to look for unique artifacts in your sequencing pipeline, reasons or flagging false positive calls. It's great for research based on what you've seen previously, because then you could go look at the pathology and correlate. And it's very important to be contributing to public resources so that we're all sharing data as opposed to hoarding it. So in general, in silico prediction algorithms work looking at a few features. So evolutionary conservation. So at this amino acid residue, how conserved is it across species? With the theory being, the logic being, if it is very conserved, then a missense mutation in this residue will be more significant. But if there's variability across species, if it's mutated, maybe it won't be as significant. Then, as we spoke about, changes in the physiochemical properties of the substituted amino acid. So checking to see how does this missense substitution differ from the baseline or from the reference. And if there is a known oncogenic variant that is different, comparing is the difference between our newly detected amino acid more or less different than the one that's known to be oncogenic. Variant location, is it in a functional domain? Is it at the 3' end of the gene? And in general, like these missense and splice site predicting tools have moderate specificity, around 60 to 80%. And there may be a tendency towards over-predicting deleterious impact. And there is varied use in cancer. So BRAF V600E is an oncogenic mutation. It's predicted to have conflicting or even benign effects when analyzed across multiple in silico algorithms. And obviously we know that it's very impactful. So it's one of the, several scenarios unique to somatic variant interpretation that, we should be aware of and exercise caution when interpreting these in silico scores. And, while they have value in assessing variant impact in the oncogenicity guidelines, they're not that impactful, and they should really only be evaluated in the context of other evidence types. And that's what's recommended by the oncogenicity guidelines in general and the AMP-ASCO-CAP guidelines as well. And multiple algorithms should not be treated as independent supporting evidence, rather, one group of in silico prediction. And there are different tools, Splice AI for splice site prediction, and VEST4, Provian, or CAD for missense variant prediction. So original research is also a way, or a way we could look into specific variants. So those could be case reports, functional studies, or larger studies like the TCGA. And when providing information and clinical annotations for a novel variant from the literature, it's important to report the nature of the evidence, the study type and sample size, the type of data. Is it DNA, RNA data? Is it tumor only or paired? And its relevance to the tumor type based on the paper that you're reading. So this was, a lot of information about different databases, and I know it's just, one thing after the other. So we're going to move on to a case and sort of watch how we would use that in real time. So this is a 75-year-old female with a sinonasal tumor. This is what it looks like from low power. This infiltrated nested lesion. Here again, we have one fragment that we'll be focusing on. Again, it's the sort of anastomosing nests or single irregular nests of cells. There appear to be some comedotype necrosis and maybe some glandular spaces towards the bottom of the tissue fragment. We get a little closer, we could see these appear very squamoid. There are some clear areas as well, and definite comedotype necrosis. Here at even higher power, I think you can notice at the bottom of the screen some mucin in the cells, some gland formation, and a focus of lymphatic invasion there in the middle. Otherwise, there's some cell clearing, but definitely a very squamoid appearance, and some vague papillary cores that appear to be coursing throughout the tissue here on the right side. So more of an endophytic inverted growth pattern. Here, there's some of the intracellular and luminal mucins highlighted by a musicarmin. So for this tumor and the location, the differential diagnosis was a carcinoma ex inverted papilloma versus mucoepidermoid carcinoma, which was considered less likely. P16 was negative. So a sample was microdissected for RNA and DNA sequencing. And this is the CCO indications for sinonasal tumors. Once you exclude viruses and SMARC deficiency, then DNA and RNA are warranted to look for fusions such as DEC AFF2, EGFR exon 20 insertion mutations, which categorize or characterize inverted papillomas, EWSR1 FLY1 fusions, which would be another squamoid type of malignancy, adenomatoma like Ewing sarcoma, IDH2 for SNUC, NUTM1 fusions for nut carcinoma, PAX3 and PAX7 for biphenotypic sinonasal sarcoma. And the DEC-AFF2 is a squamoid carcinoma that's driven by that fusion. So that's what we ended up doing in this case because of our differential diagnosis. It was either something that will be found on fusion, like mucoepidermoid carcinoma, or carcinoma ex inverted papilloma, which would be an EGFR exon 20 insertion. And this is what we found. So this was the output from our lab, and it's in that same file type that we saw before, where going from left to right, column A is the variant number, column B and C are the chromosome number and position. Then we have the reference sequence and the alteration. So an insertion of in the first row, and then a substitution in the next two, and then a deletion in the last row. QC filter pass, allele frequency would be next in G. The depth of sequencing is DP, then we get the gene, we get the HGVS codon, and then if we skip to column O, it's the HGVS protein change. The effect type or the mutation type is in K. L is the RefSeq ID, transcript ID. And then as we go more, the ClinSig number in T is the ClinVar readout, and then in X, it's the count in Cosmic. So we're going to go through one by one. So first, we're going to start with the TP53 variant, which is in row 3. So Franklin by Genox is one of those useful tools that I had highlighted earlier that takes a lot of the information and puts it all in one place in a way that's very easy to use. So I'll show you how we use this. So I put the variant up top. So you just type in either the codon change or the amino acid change. Then it prompts, are you referring to this genomic coordinate change or this other genomic coordinate change? And it gives you a bunch of variant IDs, a bunch of transcript IDs. So looking back, we're talking about the top one. So if you see the chromosome and position, um, it's the top one, which is why we say to use genomic coordinates where you can, because it allows for transferability between resources. And then also, if you take a look at our RefSeq ID, that's also listed as the canonical transcript ID in the top option. So this is what you open up to in Franklin, and you could toggle to somatic or germline interpretation. So here, there was some prognostic significance in a head and neck squames, so for tiering. And then next, it actually gives you the ACMG germline classification or the oncogenicity classification according to what it can pull from publicly or automatically. So it's not a thorough evaluation of every one of those metrics, but it's what it's able to pull. So it may not be able to pull evidence for other metrics that you could find. So it's not complete, but it is pretty good. And here you could see it's same amino acid change as a known oncogenic variant. It's a null variant. It's a termination in a bona fide tumor suppressor, and it's a non-synonymous variant in a well-established functional domain. So it has a lot of points that are going towards it, and you can look at all of these details and it's referenced out. Then you could go look at the region. So if you click on variant assessment, there's a region viewer. Looking for things like homopolymer, that's helpful because you could see the reference sequence up top. And further down, there are predictions, as you can see on the left-hand column. Fourth from the top, there's predictions, so you can actually look at in silico predictions for the specific variant. And then something that's very helpful is the publications. So there's a publications tab, and you could then look at which articles talk about this specific variant. You could then look at which talk about that amino acid, so not necessarily the same change at the amino acid, this being Q165 termination, Q165L, for example, you'd find under the amino acid tab. Then it goes out to domain and whole gene. So you could look at publications for any of these. Next, moving to OncoKB. So a truncating mutation in TP53, they sort of have categorized on their own as likely loss of function. So in summary, for those reasons, I have it as oncogenic and as tier 2. Next will be the Notch1 variant. So here, this is the view of Notch1 in OncoKB. And interestingly, Notch1 can function as a tumor suppressor and oncogene. So you have some variants that are oncogenic for loss of function, like truncating mutations upstream of a certain region. And you also have certain mutations that are considered gain of function and oncogenic, and those would be truncating mutations in a specific domain that regulates Notch1. So if you lose the regulation, you'll cause overactivity that will then lead towards oncogenicity. So another one of these useful tools is Protein Paint by St. Jude's. So here you could just search for any gene. There are lots of things you could do here, but something I use it for is looking for any gene. So in that top search bar, you just type in the gene name, you select it from a drop-down menu, and then you get a representation of this gene that you could see the functional domains of the gene. And you could try to see where your mutation is located. You could also click on, this is where it has integrated multiple resources here. You could click on a cosmic bar, that toggle up there, and all of the instances in cosmic will come up. And you can actually click on these specific mutations and look at the list in cosmic of type, organ location, tumor type, and histotype if available. And the same thing is available for ClinVar, where you could actually filter out some of these calls, some of these lollipops based on if it's called pathogenic or not in ClinVar. So another way we could look at the protein domains is UniProt. So here you could see the function of the gene, and on the left-hand side, if you click on family and domains, it will take you to the domains of this gene. And sometimes there's a little bit of a difference between protein pain and Notch1, and you may want to look at the literature to see, if anybody's done more work looking at the specific functional domain locations of specific genes. So in this gene, Notch1, it's oncogenic, it's negative from population databases, and it's a null variant in a tumor suppressor at that location. And tier 2, there's rare therapeutic reports for mutation type in head and neck squamous cell carcinoma. Next was ABL1. So in some kinases, in-frame deletions are oncogenic. So this is one that I always, would want to look into in more depth. And you can see it's a deletion of that AAG. And you can see there's a string of AAGs, AAG, AAG. And if you look at our in-house database, we see it in 11% of cases, always at a low frequency. So if it were array polymorphism, I think we'd expect it at a higher allele frequency than we see it here. So the conclusion here is that it's artifact. And the last mutation we found in this case was HER2 duplication. It happens to be an exon 20, which is the exon that is characteristic of carcinomax inverted papilloma in EGFR. So let's look specifically at this mutation. So in OncoKB, it's called oncogenic gain of function, very well studied in breast, esophagogastric, endometrial, and lung cancer. And it's in a very specific functional domain. It is a therapeutic target in different types of cancers. And here I'm just going to go through and show you the different levels of therapeutic evidence that they give you in OncoKB. So response to an FDA-approved drug in specific indications. So this would be for lung cancer. Biomarker recommended by NCCN or other expert panels, predictive of response to an FDA-approved drug in this indication. So that would be for breast, cervical, or lung cancer. Compelling clinical evidence would support the biomarker as being predictive of response to a drug in this indication, but neither biomarker or drug are standard of care in breast, non-small cell lung cancer. And in hepatobiliary cancer, it's a compelling biological evidence as being predictive of response to a drug, but neither the biomarker or drug are standard of care. So pretty happy that this is oncogenic and functional. Looking at this mutation in protein paint and specifically cosmic in protein paint, I wanted to see, okay, what have we seen it in? Because it's sort of strange to be seeing this in sinonasal squamous cell carcinoma. So mostly lung, some ovary in the cosmic database. And then I wanted to see, okay, in cBioportal, which draws from different data, looking at all head and neck studies and querying by gene. So I selected head and neck studies on the left menu, and then I clicked query by gene, and I entered ERBB2, which is HER2. So lots of mutations in head and neck cancer for HER2. But if I then go to the mutations tab, and I search for 772 in the search bar, there's no result. So this hasn't been seen in the TCGA data either. So at this point, I was sort of chalking it up to similarities in function between these two genes. So EGFR is also known as ERBB1. They're very similar in function to HER2. So, similar pathway involvement, potentially clearly oncogenic. It's at a very good, greater than 70% allele frequency. So I was happy that this was oncogenic and likely driving this tumor. But that question about the exon 20 being shared between them was still weighing heavily on my mind. And I wonder if there's anything more to that or if it was just coincidence. So looking at EGFR and HER2, knowing that they're very similar and actually looking at exon 20, you could see that region of the alpha C helix has exactly the same amino acid structure from EGFR to HER2. So that mutation that we had in this region of the alpha C helix is likely functioning in a very, very similar way between the genes that are homologous in that region. So here reported as oncogenic and tier 2 in this specific tumor type. And then that will bring us to our last case, which, shifting gears a little bit, is a liver lesion that was consistent with a GIST. It was KIT positive and S100 and keratin negative. And it was referred for our GIST panel. GIST panel covers from CCL, BRAF KIT, and PGFRA. Optional will be fusions in FGFR and mutations in NF1, fusions in NTRK, and STH assessment. So this is what the tumor looked like. And again, this is something that was circled without paying that much attention to the specific histology by me. And here we have it at the end of this core. So this is the area that was circled. Very good cellularity. And pretty nondescript. So looking at our report here, PDGFRA benign variant in ClinVar was found, and that it's a synonymous variant, not at a splice site. You can see the amino acid change is proline to proline. So this is a truly benign variant. Looking at other variants that we found across all of our indications, there was a TP53P72R mutation, which was present at 50%, and this is a very common population variant, so it's a benign variant as well. The ClinVar population is drug response. But again, that's why we don't take any one database as the be-all and end-all. So that's a benign variant. And GNA11, though, is a pathogenic or likely pathogenic variant at 14% here. So looking more at GNA11, it's recurrently mutated in uveal melanoma. This specific mutation is a hotspot. It's in the GTP binding pocket of the RAS-like domain of the protein. It's been found in melanomas, and it's activating in functional studies. So here, looking at protein paint for this specific mutation, you can see there are 329 instances in COSMIC. And if we look at them, they're pretty much all uveal melanoma or some other melanoma or melanocytoma. So all melanocytic, predominantly uveal, anastomotic hemangioma in a couple of cases. So there's a lot to go through, but this spindle cell lesion. So, okay, let's confirm what we saw. So this is DOG1, which is negative in the tumor. This is a SOX10, which is positive, and HMB45, which is positive. So thankfully, we had it on our panel due to its being covered and recommended by CCO for uveal melanoma. And this is what we suggested that this was. So just rehashing my approach now that we've seen a few cases and the databases, looking at the VAF, how does it compare to the VAF that we are validated for in our assay, and how does that relate to the tumor cellularity? Automated information from ClinVar. Are there any pathogenic or likely pathogenic calls from ClinVar that I need to evaluate before I start filtering on other bases? The variant type. Is it one that's expected to be impactful or not? Is it intronic? Is it untranscribed? These are things that I try to filter out. So then any known benign, unequivocally benign variants, I will also filter out. Then I'll filter out by NOMAD, minor allele frequency, and our in-house frequency, setting different cutoffs. And then I go on to interpret the relevant genes. And then to briefly touch on reporting. So you want to talk about the patient and sample ID. Was it, from blood, bone marrow, colon, etc.? Is it fresh, frozen? Is it a cytology preparation? Is it FFPE? The testing indication. So what is the indication for testing? In this province, it's sort of the CCO indication. Then our key findings summary. So clinically significant information. BRAF V600E mutation identified in a melanoma. Then to move on to the variant details, which would be the transcript ID, the codon change, amino acid change, if applicable, variant allele frequency, our tiering and oncogenicity, and then including if the sample passed QC and what aspects may have led to it not passing, or in what areas did it not pass, depending maybe at poor coverage of specific areas or specific regions of panel genes for this indication that you didn't find. So that you didn't have enough coverage of key genes in a negative test, that's a bigger issue than not enough coverage of a non-panel gene when you already have a variant that's significant. And then the technical details and how, including the how you prepare the specimen, how you test the specimen, and how it was validated. And so that really brings me to the end of what I had for today. I wanted to thank you all for your attendance and attention for this course and this lecture. Dr. Yael Fisher, Dr. Anna Plotkin for facilitating, and Jenny Bozek as well for helping us get everything off the ground here, and for running the course really, and the Sinai Molecular and Bioinformatics team as well. So yeah, open it up to any questions.
And I also want to thank Elan and Yael for this amazing series, and thank you for pioneering this educational event for University of Toronto. And I want to remind you that Dr. Elan Hahn is assistant professor at the University of Toronto, and he works at Mount Sinai Hospital. He's a molecular and head and neck pathologist, and Dr. Yael Fisher is a molecular pathologist from Israel who is currently clinical fellow at Mount Sinai Hospital.
Yeah, and just a reminder for everybody to fill out the survey that we're going to be sending out. Very helpful for us going forward and evaluating how we did.
And just a reminder to everybody, if you want to listen those lectures again, please welcome to our website. I really encourage you to re-listen, if you're not a molecular pathologist, to re-listen those lectures. They very, very well structured, and they provide the basics for the molecular pathology. And I think right now you have a lot of things coming, Elan and Yael.
I see one question about poly and MMR deficient tumors where they're hypermutated. They do return tons of variants. And ideally, they all need annotation. Most significant would be those driving it, though. So being aware of, a poly mutated or MMR deficient tumor, highlighting that mutation. But I think it is important to classify all of the, significant variants that we find. If you want to limit it to the, to the indicated gene list, that's sort of, lab dependent on what you report in these cases and in cases in general. And then I see, so the sinonasal case, that was a carcinomax inverted papilloma. Then there's a question about interpreting variant allele frequency. Is there a approach to determine if it's germline or somatic? It's hard to. I think you could see variation in even a germline variant that's a known germline. You could see it lower, much lower than 50%, 30% even, and higher. So I know MSK, due to their impact study, which sequences tumor normal pairs, put out a guideline for specific gene types, which are more likely to be germline rather than somatic. And variant allele frequency cutoffs, I think 30 or 35% maybe. But it's hard to say. You could get loss of heterozygosity or copy number changes that could impact these things as well.
I don't see more questions so far coming, just because it's an international audience. Reminder again to check our website for the recording. And if anyone from the Canadian or American pathologist needs CME event, you need to fill out the evaluation form to receive a CME. Okay, with that, I would like to conclude this event. I think it was a huge success, and thanks again, Dr. Hahn and Dr. Fisher, for your amazing work. And we'll see you all guys at other events, and please follow our U of T CPD website. Thanks a lot, everybody.